It's my pleasure to introduce Dr. Doug Lowy, who is the Acting Director of the National Cancer Institute, and Dr. Tom Misteli. I will be moderating. My name is Chris Heery. I am a medical oncologist. I work with the Laboratory of Tumor Immunology and Biology. I started at the NCI in about 2009, did my medical oncology fellowship here, and was lucky enough to stay on working with two great mentors, Dr. Jeffrey Shlom and James Gully. This is an area of interest for me, how basic research, how the process works. I work in the clinic, and so we work on the direct result of things that come out of basic research. So with that, let me first introduce Dr. Lowy and let him say a little bit about himself, and then Dr. Misteli. Dr. Lowy first.
Good morning, everyone. I'm Doug Lowy. I've been the acting director of the National Cancer Institute since April, but I have run a basic research laboratory at the NCI since 1975. I have also been fortunate to take some of the discoveries that we have made in the laboratory and to develop them into an intervention, which is the HPV vaccine. But in the lab, John Schiller and I and our colleagues developed the technology that led to the vaccine. But of course, as with other interventions, they are developed really by the pharmaceutical industry rather than by the government. I'm looking forward to this discussion about the relevance of basic research to the overall area of cancer research.
Great. Dr. Misteli?
Okay, good morning. I'm Tom Misteli. I'm a senior investigator at the Center for Cancer Research here at the NCI. I guess compared to Dr. Lowy, I'm the new kid on the block here. I've only been here for just over 15 years. I've spent my entire independent career here at the NCI, and you know the NCI is a tremendous place to work. And I can see how a few years turn into a few decades here. So I understand Dr. Lowy's situation. So I'm a classically trained cell biologist, and my laboratory studies what is probably one of the most fundamental processes in biology, and that is the question of how genomes are organized in three-dimensional space inside of human cells, and how this organization affects the function of genes and genomes, both in healthy cells and in disease, obviously cancer. And what's been particularly fascinating for me over the years about this topic has been that we're starting really all our studies by addressing very, very fundamental, very basic questions. But it turns out that just about anything we find has some sort of implication for clinical applications or practical applications. And for me, as a basic cell biologist, that's really been eye-opening and very exciting.
Thank you, Dr. Misteli. That was fantastic and really interesting to hear how these things can be applied going forward. If you have more interest in the topic and more interest about learning about the National Cancer Institute, please visit www.cancer.gov. Now we're going to move on to the question and answer portion. And if you have additional questions as you're watching and you'd like to send them in, please on Twitter use the hashtag AskDrDoug with no period. So it's AskDrDoug. You can see it up above Dr. Lowy's head there. So we'll start with our first question. What is basic science? So a nice broad question for Dr. Lowy to start with. What is basic science?
Basic science is really a wide spectrum of research areas, but in the context of cell biology that Tom Misteli was talking about, it really has to do with trying to understand the fundamental processes of biology. And this can be looked at the level of an entire organism, for example, in the mouse or in the fruit fly. or even in a slime mold or something like that. And remarkably, there are similarities between all of these different organisms and even among bacteria in terms of fundamental processes that regulate biology. Within the context of cancer research, virtually any area of the regulation of cell growth, cell viability, and signaling between one molecule and another can affect these fundamental cellular processes, and not infrequently they go awry in cancer. And that's why there is such a close correlation between the study of basic research and its potential application to the clinic, as Tom Misteli was mentioning.
Great. Dr. Misteli, do you have anything to add to that?
Yeah, I just want to echo what Dr. Lowy said. I think basic research is really the foundation for all practical applications in biomedicine. And I think, you know, this is also true not just for biomedicine, but for any technology. You know, the cars we drive, the iPhones we use, the computers we use, they're really all based on basic research, and that's not different in biomedicine. Because if you think about it, you know, the end product of biomedical research, the ideal end product of biomedical research is a drug that cures a disease. But it's very likely that a basic scientist identified a potential target for a drug by studying the difference between a cancer cell and a normal cell and noticed that something is different. And then a chemist was probably involved in actually synthesizing the drug. And then you have to deliver the drug, and that involves knowledge of how molecules get into cells. So every single step of a therapeutic approach really is based on basic science. And I think that's important to keep in mind.
Great. Those were wonderful answers. Thank you both. We're going to move on to the next question then. And maybe this time we'll start with Dr. Misteli. The next question is, how is basic science applied in cancer research?
Well, I think, as I just said, I think every aspect of cancer is really subject to investigation by basic scientists, because we just need to understand the molecular mechanisms that cause and drive cancer. And without that knowledge, it's very, very difficult to develop specific therapies, targeted therapies, and effective therapies. And also, if we don't even talk about therapies, just starting with diagnosis. Of course, we can do fairly superficial diagnosis based on morphology of cells or what have you, but those are usually not very accurate diagnostic tools. So the more molecular we can go with our diagnostic tools, the better. And again, what that means is that we need to understand the underlying molecular machinery that's different in cancer and in normal cells.
Dr. Lowy, do you have anything to add to that?
I would like to echo what Tom has been saying. We know of so many breakthrough findings that have been made through basic research. For example, the study of genes that promote cancer, so-called oncogenes, genes that inhibit cancer, tumor suppressor genes, genes that affect the viability of cells, that make cells either live when perhaps they should not be doing so, or make them die inappropriately. The regulation of the immune system. All of these different areas play upon the development of interventions and also our understanding of how cancer comes about. And as Tom mentions, as a result of understanding the underpinnings of the abnormalities, it has become progressively more possible to develop interventions that are able to overcome these abnormalities. So I think that there is this very close and direct relationship between basic findings and the development of interventions. These interventions can be working at the level of prevention, screening, diagnosis, or treatment. And in all of these areas, we really are grateful for the advances of basic science that have made the insights possible that have led to these interventions.
Thank you both. Now, the next question continues sort of, on what both of you have already touched on a bit, but really asking how the NCI plays a role in this. So the question is, what does basic science mean specifically to the NCI's role in cancer research? And I think, Dr. Lowy, maybe you could start with this one.
Yes, the NCI places great value on fundamental research that uncovers these areas of basic biology and also host defense mechanisms against the development of tumors. And we support a wide array of basic research in many different areas. The vast majority of that research is proposed by investigators, so-called investigator-initiated research, and this is for us a really critical area of support. One of the things that has happened in the last few years is that because of the hyper-competitive nature of the funding mechanism, we have been concerned that our investigators may not be taking as many chances as they otherwise might take with their basic investigations. And so we have developed the Outstanding Investigator Award, which we piloted this past year. It is for investigator-initiated research. A large amount of it is basic, although it can also be applied. But instead of being a 5-year grant, the award is made for 7 years, and the size of the award is substantially larger than that of a usual R01 or a major research award. And our expectation is that further breakthrough findings in the basic area as well as applied will be coming from these investigators. I only highlight this as one new example. We continue to rely on basic research conducted by our investigators through more traditional grant mechanisms as well.
Dr. Misteli, do you have anything to add to that?
Yeah, I think one thing that's important to keep in mind is that an important hallmark of basic research is that one never quite knows where the next breakthrough is coming from, which parts of basic research will all of a sudden become important for a particular disease, a particular cancer type. And one of the benefits, I think, of the NCI, and this is true both for our intramural research program here in Bethesda, but also the extramural program, is that we really span the range of scientific activities. Just as an example, here on the Bethesda campus at the NCI, we have biophysicists who study how proteins fold inside of cells. And we also have people who do actual brain surgery. So we have the entire spectrum. And I think this is really important because of this uncertainty of where the important advances will come from. And so we really need to cover all our bases in a sense. And I think the benefit of the NCI being a fairly large institution is that we can actually do that. And I think we try in our portfolio to be very broad and cover all possible bases.
Thank you. The last question of our pre-planned questions is, what role did basic research play in the development of HPV vaccine? And I know we were switching back and forth, but I think that this one might be best directed to Dr. Lowy.
But the role of basic research really was first to develop molecular cloning technology, which made it possible to isolate individual HPV genomes. And then Harold Zerhausen and his colleagues at the German Cancer Research Center set about testing various cancers to see whether he could find HPV DNA in those cancers. And this led to the discovery of HPV 16 and 18 and their role in the development of cervical cancer and the eventual demonstration that virtually all cervical cancers are attributable to HPV infection. This finding also has been expanded in a number of different ways. Perhaps the most important is to identify other cancers that are also caused by HPV infection, especially by these two HPV types, 16 and 18. This means the vast majority of anal cancer, a high proportion of vulvar and vaginal cancer, and particularly epidemic in the U.S. and many other industrialized countries are oropharynx cancer. So that the basic research really has identified the wide spectrum of the disease where these viruses are found. In addition, the pathogenesis of these viruses has been understood in much greater detail as a result of their isolation and study. And in addition, trying to understand the natural history of HPV infection and its role in the development of cervical cancer and the other cancers. All of this information has essentially then come together in the areas of cancer prevention, primary prevention with the HPV vaccine as a result of understanding the underlying biology and when it is most appropriate to intervene, and understanding the development of neutralizing antibodies by the virus. It was possible to develop an effective HPV vaccine, which leads to the primary prevention of HPV infection and the downstream precancers and cancers. In addition, understanding the cause of cervical cancer has led to what I like to think of as etiology-based screening. That is, we are moving away from the primary screening modality being cytology or the pap smear, which is very good, but even better is using HPV-based screening. And in addition, there is current research that is trying to refine HPV-based screening through ancillary tests, which are based on our further molecular understanding of the pathogenesis of cervical cancer, so that we can end up with screening that leads to the most benefit and the least amount of harm. Thank you.
Dr. Misteli, I don't know if you want to add anything to that, but if you do, feel free.
No, I think that was well put.
Great. So now we have the opportunity to move on to questions from Twitter. For anyone who has posted a question, if we do not get to your question, please know that we will try to answer it via the NCI Twitter feed. But we'll try to get to the ones that are sort of most applicable to this main topic of basic science. So if you asked a question that's not really on this topic, we are going to focus on those that did. The first one comes from Dana Farber, and they ask, how does the NCI decide to allocate its basic research funding, and where are the priority areas?
The basic research funding, by and large, is determined primarily by the research study sections of the NIH. And these research study sections are populated exclusively by extramural investigators, and they rate the applications. The NCI does prioritize to some degree, but the vast majority of the allocation is really determined by the study section evaluations.
Dr. Misteli, do you want to add anything to that?
Well, maybe just to add that, yeah, I think an important hallmark of basic research is that, in a sense, it's a bottom-up approach. Basic research is really driven often by individual investigators making a observation, being intrigued by an observation, and then following that up. And I think, as Dr. Lowy said, I think the hope is that the most intriguing observations will make it through study section and will be picked up in study section. And so in that sense, it's more of a bottom-up approach.
Great, thank you. So I'm going to try to combine two questions here. So I'm going to ask both of them together because I think they fit nicely together. The first one comes from Cold Springs Harbor, and they asked, so much is left to discover about cancer. How do we decide when basic science is ready to transition to applied? And then another question, which is along the same lines, comes from Jennifer Windrum in Omaha, Nebraska. And she asks, what does basic science research mean for patients who currently have advanced cancer? So I guess the question that I'm trying to put together is here, how long does that transition take? How rapidly can we see these things develop into practical applications, and how do we let that guide what's happening in the basic science setting? Dr. Misteli, do you want to start with that one?
Sure. Well, first of all, hello to Cold Spring Harbor, where I did my postdoc. So I think this is obviously a critical question. How do you translate, in that sense, basic research into the clinic? And again, a lot of it has to do, I think, with bringing the right people together. Because it is very difficult for a basic scientist to drive their work all the way into the clinic without the help of clinicians, obviously. And I think, conversely, it's difficult for clinicians to go all the way from very basic research to the clinic. And so I think the key is bringing people together, people from different disciplines, basic scientists and clinicians. And I think each institution should, at a local level, make an effort to do that, to pair up basic scientists and clinicians and generate mechanisms whereby they don't live in separate parts of the institution and can collaborate. And I think it's difficult to sort of give an average time of how long it takes for a basic discovery to be translated into clinic, because, as I said before, often the relevance of a basic discovery will not become clear or is not obvious until, for example, a clinical discovery is made and the two are then matched. And then sometimes these things can move fairly rapidly. So it's, in a way, a case-by-case issue of how long it will take and how exactly the process works. I think it's all about collaboration between basic scientists and clinicians.
I would essentially agree. We need to continually be concerned first about what is the clinical need, but at the same time, what are the clinical opportunities? And we usually try to err on the side of trying to do things that are hard, and to try to make progress go faster if we possibly can. But it is not straightforward in cancer research to go from a basic finding to an applied one. Sometimes we are very fortunate and an abnormality is identified in a range of cancers for which there might already be a candidate drug. And under those circumstances, the application can be very fast. But in most situations, the basic finding and its translational to clinical application takes a fair amount of time. And as Tom Misteli says, it really tends to be a team sport with a range of investigators with at least partially non-overlapping expertise banding together to make progress towards the goal of developing an intervention.
So Dr. Lowy, a question that is similar and along the same lines is asked again. This one's by Dana Farber again. So they asked, what is an example of a basic science finding that has led to a clinical impact, and how long did it take? And I thought maybe you could use the example of HPV to walk people through just how long that took.
Well, it depends on where you start with HPV. The identification of HPV in cervical cancer and other cancers was made really in the 1980s. The technology for the vaccine was developed in the 1990s, and the clinical testing and validation of it being effective took place in the 2000s. So you could argue that probably a 20-year period from the initial basic discovery to application. When it comes to treatment, there are some situations which have developed much faster. For example, finding mutations of the BRAF oncogene in melanoma was identified in the early 2000s, and the treatment of BRAF targeted treatment was approved less than 10 years later.
Right. Dr. Misteli, do you have any thoughts on that?
Yeah, so I'm going to go out on a limb here and say that I actually think we're going to see faster translation of basic observations into the clinic in the future. And the reason for that is that we are accumulating a tremendous wealth of basic knowledge. And in parallel, we're identifying disease genes at an increased rate. And as I said before, it's really a matching process in a sense, because of course when a disease mutation, be it cancer or any other disease, is identified, the first question is, well, what does the gene do? What does the mutated gene do? And more and more, we can simply tap into this vast knowledge of that basic science has created on sometimes obscure-looking genes or proteins, but people have spent decades working on and just trying to understand the basic mechanisms. And if those genes turn out to be disease genes, then we can use that knowledge immediately and work towards treatments. And so I think we're going to see an acceleration of that pipeline over the next years or decades.
Thank you for that. That's some great insight. I guess this is a little off of the Twitter questions, but since I'm asking, I'll go ahead. Do you see any, along those same lines, any increasing speed of translation as a result of basic discoveries that have gone into the clinic and then findings from the clinic that are applied back in the basic lab again, and that transition back and forth between the two? Dr. Lowy, could you comment on that?
Certainly. We see that there are discoveries going from bench to bedside and then from bedside back to bench. And I agree with Dr. Misteli that the pace of the discoveries and applications appears to be increasing. but it still takes a fair amount of time to go from a basic discovery to application. Among other things, you need to undergo rigorous clinical trials to demonstrate that the benefits that accrue from a particular intervention actually merit the FDA approval and that the side effects are acceptable.
We've had a lot of questions about both through Twitter about both immunotherapy and pediatric cancers, and so I'm going to ask first about pediatric cancers in a broad way. Could you comment on the NCI's role in advancing basic science research in pediatric cancer, and what the NCI's role is in allowing those advances to get closer to the clinic?
The NCI supports research both in adult as well as in pediatric cancers, and the basic research really is in both of these areas. For example, we have supported research that demonstrates rearrangements of genes in a wide variety of children's cancers, and we are in the process of trying to see whether these kinds of observations can now be applied for treatment. So that's what happens in the basic area and its relation for pediatric cancer. We also look at the germline of patients with pediatric cancer to understand the predisposition that they may have to the development of these cancers. And in the long run, we hope that these insights are going to lead to better interventions. In the area of adult cancer, for example, germline mutations involving the BRCA genes we know lead to predisposition of breast cancer and ovarian cancer, and quite recently the first FDA-approved intervention for the treatment of ovarian cancer for patients with mutations of BRCA has been FDA approved, and this is based really on a molecular understanding as well as the predisposition of these patients to develop the ovarian cancer.
Dr. Misteli, do you have anything to add to that?
Well, maybe just to say that pediatric cancers can be particularly insightful because they are different from adult cancers, and so I think it's one of these cases of comparing different cancers, comparing the mechanisms between pediatric and adult cancers. I think again is in a way a tool to make progress and actually get at cancer mechanisms. And I think that the same can be said for rare tumors, which are another very well-defined and I think interesting subset of tumors, which we are at the NCI very interested in. And for the same reason that it provides a fairly well-defined group of tumors, and we can study them in a very focused manner. And again, the hope is by doing that to get at molecular mechanism.
Chris, I did want to try to address the issue of immunotherapy and immunology. The fruition of immunotherapy right now is a result of basic research in trying to understand the fundamental mechanisms of host defense and the immune system, as well as understanding of cell surface proteins, etc. And the recent development of FDA-approved checkpoint immune inhibitors really is the outgrowth of many years of fundamental research. But in addition, there are other interventions which have considerable promise, and they also are based on a fundamental understanding of how the immune system works and trying to harness it for the treatment of cancer. and so I think that this is a beautiful example where basic research is now paying off. But once again, it took a long time from the basic findings to their clinical, practical application.
Thank you. Yeah, I think that's always a good point to reinforce, that many of the discoveries that have led to these clinical trials have been many years in the past. Well, I think we have answered almost all of the questions that we can get to. We're a little over the time that was allotted, so I would like to just thank Dr. Lowy and Dr. Misteli for the fantastic insight they provided today, and encourage any viewers who might be watching this on delay. You can still communicate with the NCI through Twitter, and you can also get more information about many of the topics we've discussed on cancer.gov. With that, I think we will stop the broadcast, and thank you for your participation and viewing this first ever NCI Google Hangout.
Thank you, Chris.
Thanks so much, Chris, and Tom as well, and the people online who asked interesting questions.
Thank you.